Hi, this is Mrs. Lockett. This is Unit 7, Notes 2 on bacteria and viruses. Okay, so the first thing that we're going to do is we're going to classify our prokaryotes, and this goes on from what we talked about last time, and that is that there's two different groups of bacteria, the eubacteria and the archaebacteria. Those are the names of the kingdoms. They belong to domain bacteria and archaea. And the differences between them include that prokaryotes live almost everywhere, and they are surrounded by a cell wall with peptidoglycan, and cell walls just protect them from injury and determine its shape. And the most common bacteria that you would probably be familiar with is E. coli. Our archaebacteria are also prokaryotes. They lack peptidoglycan. They have a DNA sequence similar to eukaryotes in that they have histones that wrap their DNA. They live in extremely harsh environments, so they're called extremophiles. And let's continue on talking about bacteria. Alright, so here's just a couple pictures of eubacteria and archaea. And I've got, let's see, I'm pretty sure this is an E. coli. And as a matter of fact, I'm positive that's an E. coli. And this is a methanogen, which is one that lives in cows and makes methane. Alright, so this is a typical eubacterium. So you have this is all review, but cytoplasm, ribosomes, you've got the DNA, which is the nucleosome. You've got the cell membrane, you've got the cell wall, which I believe is the red line, and then you've got a capsule, which is yellow. I am messing up all over the place today. And then you've got pili and flagella all sticking off of the sides. Alright, so we identify prokaryotes in 4 different ways: by shape, by cell walls, by movement, and by metabolism, how they obtain their energy. So we're going to talk about shape first. There's 3 shapes that you need to be familiar with. One is called cocci or coccus, and they are round. And then you have bacilli or bacillus, and they are rod-shaped. And then you have spirilla or spirillum, and they are spirals, so they twirl. The cell wall can be, they're made out of peptidoglycan, and so the difference is that you've got gram-positive and gram-negative. Gram-positive are going to stain purple, so and then gram-negative are going to stain pink. And the thick walls of the gram-positive hold onto that stain, which is why they stain purple, and the gram-negative ones have an outer lipid layer that kind of blocks that stain, so they end up being pink. Alright, prokaryotic movement can be that they have no movement, that they have flagella that make them swim, or they have cilia that make them swim, or they can lash, which is basically kind of like a snake movement. They can snake, like they can slither, and they can spiral forward where they are twirling. Prokaryotes can be heterotrophs, where they take in food, and there's 2 different kinds. You can have chemoheterotrophs that take in energy and carbon, and you have photoheterotrophs that are photosynthetic, using sunlight for energy, but they also take in organic compounds. Autotrophs can be photoautotrophs, where they use light and photosynthesis to convert carbon dioxide and water into food, or you've got chemoautotrophs that make organic compounds from carbon dioxide. For growth and reproduction, they have 3 ways of dividing. Number 1 is binary fission, where the single cell separates into 2 cells by division. And if you watch the little GIF there, you can see, let me move me so that I'm not blocking it in case I am. And you end up with clones of the original cell, and this is asexual reproduction. You have conjugation, which is where bacteria are exchanging genes between cells. This is that same as that lateral gene transfer that we had in the last unit. And it increases diversity and is considered sexual reproduction because you are exchanging genetic material. And the last one is spore formation, which is a protection that they can undergo to protect themselves in unfavorable conditions. Like if they if they require moisture and it dries up, they can make spores so that they'll survive. All right, here's just another picture where you've got binary fission, so you've got two identical cells. Conjugation, you can see the little tube that goes from one to the other. And spore formation, all of these different little things are spores. All right, the importance of bacteria is that they are decomposers. They're going to recycle organic material. They're breaking down dead organisms. They're going to be able to treat sewage. So they do things like that. Humans have harnessed their power in order to make food and beverages. We use them to digest petroleum, like for oil spills. We use them to remove waste materials from different things, and we use them to make drugs and chemicals. Like, remember when we talked about our biotechnology, and we use E. coli in order to make insulin. And then finally, you have symbiotic relationships. These are very important. Bacteria make vitamin K in our guts, so we can't make vitamin K, but we need it, and they make it for us, so that's a good thing. They control colonization by pathogens. You have to have good bacteria in your gut and on your skin in order to keep the bad bacteria from taking over. And plants are super important for nitrogen-fixing bacteria because our plants are how we get our nitrogen, and plants can't get nitrogen in the N2 gas form. So we have these great bacteria that are going to convert N2 gas into nitrates and nitrites that plants can use. All right, now we're going to talk about viruses. And so viruses are particles of nucleic acid, and they have protein covering them and sometimes a lipid on the outside of that. Super important thing to remember is viruses are not living. All right, they can only respond by infecting living cells, or they can only reproduce by infecting living cells. All right, so here's just 3 examples of some viruses. Let's see if this, yes it does. All right. Okay, so you've got the T4 bacteriophage, kind of looks like a little alien ship. Those are the ones that infect bacteria. And we talked about these when we were doing biotechnology and DNA as one of the ways that they use to discover what genetic material was. There are other ones that are kind of capsule-shaped. This is the tobacco mosaic virus, which infects plants. And then we have this one, which is kind of like a coronavirus, but it's an influenza virus, and it's got RNA as its genetic material. It's got a capsid, which is the purple, and then it's got a lipid envelope, and it's got surface proteins. And we're going to talk about another one just like that in a moment. All right, so the genetic material for viruses can be DNA or RNA, just depends on what the type of virus is. They get protection from their protein coat, which is called the capsid. And the capsid is important because it enables the virus to get into the host, and then once the virus gets inside, it's going to transcribe and translate its own genetic material using the ribosomes and enzymes of the organism it is infected. And viruses are very specific to the cells they infect. So viruses can't you don't have viruses that infect everything. They are very specific about what cells. It depends on the receptors on the cells. All right, here's a picture of a coronavirus, and this is, of course, the one that causes COVID-19. And just so you have some knowledge about this, it is an RNA virus. It has a capsid, it has an envelope, and then it has the glycoprotein spikes that you can see all surrounding this. So that is the one that is causing all the issues these days. All right, when you are infected by a virus, you can get 2 different types of infection, lytic infection or lysogenic infection. So in a lytic infection, the virus is going to take over the cell. It's going to make copies of itself, and it's going to cause the cell to burst. So you can see in this picture, this is a bacterium. There's the T4 bacteriophage. It has inserted its DNA. The DNA is going to come. It's going to reproduce. It's going to start using the ribosomes to start making its protein capsids. And now I've made all these copies of myself, and I'm going to actually bust out of the cell so that I can go and infect more cells. So that's all I want to do. All right, a lysogenic infection is when the virus integrates its DNA into the DNA of the host cell. And so you can see that I have, again, a bacterium. This is a T4 bacteriophage. It's inserting its little DNA there, and its DNA is going to join in to the DNA of the host cell. It will reproduce as long as the host cell keeps reproducing, and then eventually something will trigger it to go into the lytic cycle. All right, retroviruses have DNA as genetic material. They must infect the cell. They make DNA copy from RNA. They use reverse transcriptase to do that. And then they can remain dormant for some time. And then they might trigger the lytic cycle. HIV is a retrovirus. It's probably the most common, well-known retrovirus, and it results in the disease AIDS. All right, so this is now general stuff about pathogens, things that make you ill. This is going to be about bacteria and viruses. So some bacteria that cause illness by destruction of cells or by producing toxins that poison the host. So tuberculosis is one that destroys cells. Scarlet fever and strep throat, I believe, both make toxins. I know strep throat does. So those are things that can make you ill. And then viruses attack cells or change cell growth patterns. So polio is going to be one that attacks neurons and so causes neurodegenerative disease. The papillomavirus is one that causes warts in humans. And if you're familiar with the HPV vaccine, it is to treat papillomavirus so that men cannot give and women do not get cervical cancer. We control bacteria by heat sterilization, disinfection with chemicals, temperature, like refrigeration will help control bacteria. There's vaccines for a lot of the bad disease-causing bacteria. Use protection, and antibiotics kill bacteria. So because antibiotic means anti-life, and bacteria are alive, and so we can kill them. We control viruses in a lot of the same ways. We have vaccines. Hopefully we're going to have COVID-19 vaccine really soon. We have sterilization. So we sterilize things with high temperatures. We use protection. Protection has become a huge important thing now with our PPE, and so we've got our masks, and we have our social distancing. Wash your hands, wash your hands, wash your hands. That's good for both of them. And antibiotics do not work on viruses because viruses are not alive, and antibiotic means anti-life, so they don't work on that. There are antiviral drugs, but they are not as effective. Okay, that's it for this one. Thank you very much. And if you have any questions, please bring them to class.